Hi, it's Mr. Mazurkewitz, and in this video we're going to be taking a look at mutations. Now, mutations are any changes that I make to the genetic information, or in other words, the DNA. Now, if you know how DNA works, DNA, remember, is going to be transcribed or written down into messenger RNA in the nucleus, and then we're going to send that messenger RNA to a ribosome to be translated into an actual protein, and the protein is going to determine your physical characteristics or your phenotype. So in other words, any changes that I make to the DNA, that's going to be passed on to the mRNA when we transcribe, and then that could cause a change to my protein during translation. So our lesson essential question today are going to be, what are the different types of mutations? So we'll see that there are some small-scale mutations that exist and other large-scale mutations. And then also, how can they affect the expression of genes, or in other words, the phenotype, the protein that we make from that DNA. So before we get into them, I just wanted to start with an analogy, because your DNA is kind of like a recipe on how to make you. So in a way that if we change the recipe, we could or could not change the outcome. So here's a recipe for grandma's famous apple pie. So here are all the ingredients, and if you follow this exactly as it's written out, you should get the same result every time. You should get that delicious apple pie that grandma came up with. But what if I start making changes to this? So I'm going to make a change to somewhere in this recipe, and I want to see if you can catch it. So ready? Did you see that? If not, let me go back, and I'm going to go ahead and give you a hint. It's in the second paragraph there. I'm going to change it right now. So what you might have noticed is I changed the letter U in the word pour to an O, so now it's P-O-O-R. Now, I changed the recipe. I did change something. Is that going to change my end result? And you're probably thinking, no, if I got this recipe, I would understand what you meant, and I could create that same apple pie. And just like that, some mutations we're going to notice don't always cause a change to the phenotype. What about if I make a change somewhere up at the top? So in this first paragraph right here. Now I change from 3 tablespoons of flour to 9. Now that's probably going to cause some sort of physical change to my apple pie. It's probably going to come out worse because I'm adding 3 times as much flour. How about if I change another one here back in the second paragraph right here? Now I change from 8 sliced apples to 9 sliced apples. So I'm going to change the result, but did I ruin my pie? Probably not. You probably really like apples if you're eating apple pie, so the more apples, the better. So in this case, it was a good change. And I can also make a big, large-scale change. So let's say I change the entire recipe where the second and third parts swap with each other. Am I going to get the same result? And you're probably thinking, no way. If I bake before I create my crust and add the apples, it's probably going to come out really bad. So just like we made some small and big changes and we got good results and bad results, the same thing can happen with mutations. So the first type of mutations we're going to look at are things called gene mutations. Gene mutations can also be called point mutations because these are going to be changes that we make at a very specific point or a specific gene in the DNA. So here's a picture or just a strand of DNA, a particular gene that we're going to look at. And remember that only one out of these 2 strands is going to be used for creating my protein. So let's go ahead and get rid of that bottom one. So let's say this one here is my coding strand. Now to create my protein, I first have to transcribe, so I'm going to turn it over to mRNA, and then I'll use that codon table in the bottom right to translate it into a series of amino acids, and what you would get is the amino acid sequence leucine, glutamine, and valine. So let's say that's my normal gene or my normal protein. Now any gene mutation or point mutation is going to be changes that I make to some of those letters in this particular gene. So let's first take a look at one type of gene mutation known as a substitution. Now, if you know what the word substitute means, it means to replace with something else. So let's say I'm going to take my second A here and the GAT, and I'm going to substitute it or change it to a G. Now, how might this affect my protein? Well, now when I transcribe this, I'm going to get a change in my second letter to a C now. And now when I translate that, my protein is going to come looking like this. Now it's going to be proline, glutamine, valine. So in this case, I caused a change to my protein, which it could be good, it could be bad, it depends, but this does result in a phenotypic change. Why? Well, because it resulted in a change in my amino acid sequence. But not all mutations have to cause a phenotypic change to the protein. So for example, let's go back to the original piece of DNA, and let's do another substitution. I'm going to substitute the letter T, the third one letter here, and I'm going to substitute that with an A. Well, now when I transcribe it, again, I'm going to get a different third letter, but when I translate it, let's see what happens. I still get the original, leucine, glutamine, valine. So even though I changed or substituted a letter, I didn't get a phenotypic change. You'll notice that that codon table, some amino acids can be coded for by multiple codons. So not always is it going to result in a phenotypic change. It has to cause a change to the protein structure or the amino acid. Now, another type of gene mutation, or other types, are things called insertions and deletions. And they're exactly what they sound like. So with an insertion, it means that I'm going to insert an extra letter. So I didn't substitute it, I inserted in there. And deletions means I'm going to maybe forget to put a letter, so I deleted it from my DNA. Now, these are usually going to cause catastrophic changes or big changes to the protein. And the reason being is think about how we read DNA. Remember that DNA, or the genetic code, is read 3 letters at a time, or as codons. By inserting or deleting, how does that affect the rest of the protein? Well, it's going to shift all my letters over either to the right one or to the left one. So we call these types of mutations, insertions and deletions, they're referred to as frameshift mutations. So if you're not quite following along, let's use this an analogy of a sentence here. So I have this sentence written with 3-letter words, and it reads, "The boy saw the tan dog run." So it's a sentence made of 3-letter words. Now, if I do a substitution, let's go back to that one. Let's say I substitute the B to a T. So now how does this sentence read if we read 3 letters at a time? It says, "The toy saw the tan dog run." So even though I changed one letter, it only really affected that one word, boy to toy, but the rest of the sentence was unaffected. But what about if I insert or delete? So let's insert a letter instead of substituting it. So I'm going to add a W in between the B and the O. Now if we read this 3 letters at a time, it says the, and then you'll notice that the word boy, it's actually BWO, now YSA, now WTH, now ETA, and the rest of my sentence has been impacted. So by inserting a letter, it shifted all my letters over, so now the whole protein in this case would be messed up because all the codons have been shifted. Same thing when I delete. So if I were to delete, let's say, the B in boy, now if I read this 3 letters at a time, it's the OYS, OYS, AWT, HET, the entire sentence again becomes impacted. So we call insertions and deletions frameshift mutations because they're going to shift all my letters over once. And these usually are going to have a big catastrophic change to my protein because none of the amino acids are going to come out the same. Now, up until this point, we've only talked about these small-scale gene mutations. And remember that a gene mutation is only impacting one gene and therefore one protein. But what about large-scale mutations that impact many genes at once? We call these large-scale mutations chromosomal mutations. And remember that a chromosome is just tons of DNA that's been wrapped up and wrapped up and wrapped up. So any change that I make to a chromosome is going to impact hundreds, if not thousands, of genes all at the same time. So with a deletion chromosomal mutation, that's going to be when I accidentally leave off part of the chromosome. So maybe during replication, a whole chunk was wiped out. Now I'm going to be missing all those genes, and therefore I can't make all those proteins. That's going to have a huge negative consequence. During duplication, it's exactly what it sounds like. We're going to accidentally take a chunk of DNA and copy it twice. So now we're going to be doubling up on all of these genes that are on that part of the chromosome. With inversion, to invert means to flip upside down. So during that S phase of interphase, when we're copying the DNA, part of that chromosome might accidentally get flipped upside down. So now the genes are located in the wrong spot on that chromosome, which again is going to have a negative consequence. And then finally, the last one is translocation. And to translocate just means to locate it on a different chromosome. So here you have 2 chromosomes being shown, let's say chromosome number 4 and 20, and they're not homologous, so they code for different things, and part can break off and swap with the other. So now you have genes on those chromosomes that are located in different spots, and now that's going to cause all sorts of issues when you try and do transcription and translation. So the thing with chromosomal mutations, again, is that you're impacting hundreds, if not thousands, of genes at once, and these are typically going to have huge negative consequences. They're not going to be something that just impacts one protein. You're talking about tons of genes being impacted all at the same time. The last thing that we're going to take a look at is how do we get mutations to pass on to the next generation. Now, regardless of which type of mutation we're talking about, whether it's a chromosomal mutation or a gene mutation, remember that mutations can have all sorts of impacts. Some of them will be hurtful towards the organism, some of them will be neutral and not have any impact on the organism, and in some cases they'll even be beneficial where they'll actually help the organism survive. And we'd hopefully want to take these beneficial traits and mutations and pass them to the next generation. Now, when it comes to your cells, you have 2 major types. You have your somatic cells, which are pretty much all the cells in your body with the exception of your reproductive cells. So these would be things like your heart cells and skin cells and brain cells. Now, if a mutation occurs in one of these cells, will you pass that on to the next generation? And the answer to that question is no. Remember that your heart cells and skin cells, those are yours and yours alone. So whatever happens in those will die with you. In order for mutation to be passed on to the next generation, it has to be present in what are called the germ cells. Germ cells are the ones that will eventually form your sex cells, so sperm and egg, and that will ultimately be passed on to your offspring. So if a mutation occurs in a somatic cell, it does not get passed on, but if it is present or occurs in a germ cell, that ultimately will help it get passed on to the next generation. So at this point, make sure that you guys are able to differentiate between gene mutations versus chromosomal mutations, knowing all the different types and how they can impact the expression of that gene, and then also how mutations can get passed on to the next generation. Thanks for watching.